So I'll just start the webinar now. So hi everyone. Thank you for joining the webinar today entitled Biomanufacturing 4.0, a new era in cell and gene therapy development, hosted by BIS Research. My name is Stuti. I'm a senior research associate in healthcare cell and gene therapy, pharma tech vertical with BIS Research. And I'm really very excited to be moderating the event as well as joining as a speaker. So to begin, we are first going to go through a round of introduction to our panel. First up, we have Dr. Likhesh Sharma, who leads Cytiva's cell therapy business as a product manager in South Asia. He has more than 15 years of experience in the field of biotechnology. His expertise ranges from protein engineering and characterization to cell analysis and cell therapy. Next, we have Mr. Mario Bellman, who is an application specialist and technical representative of Enforce Latam. He has more than 20 years of experience in biotechnology with a special focus on the cultivation of microorganisms and mammalian cells. He was also a part of team of experts and product specialists at Sartorius, Mexico. Lastly, we have Akash Mahaskar, principal analyst leading the cell and gene therapy pharmatic vertical in BIS. He has about 8 years of experience in healthcare market research and consulting. He has experience in pharma and medtech. He has experience in pharma and medtech, and he specializes in competitive analysis, epidemiology modeling, and impact analysis. So that is our esteemed panel. So thank you so much, everyone, for joining us today. So before we start the event, Akash, are you presenting your screen?
Hello, am I audible, Stuti?
Yeah, you are audible, but your screen is not presented.
Uh, I
I
have
can see the
present.
screen.
I think it's
From my
present.
end, it's not visible.
Okay.
So
Okay.
yeah.
So since it is presented, I think I'll move ahead then.
So yeah, I'll just continue and then you can start. Sure. I guess there is some glitch from my end. So before we start the event, I want to run you through a few housekeeping items. So the session will begin with all the four speakers presenting their views on the topic, followed by a 10 to 15-minute Q&A session. I request the audience to post their questions into the chat box and I relay them over to the panelists. The webinar will be available to the registrants shortly after the event, and the recording of this webinar will be posted on our YouTube channel. So without further ado, I'm going to turn it over to Akash Mahaskar, who is going to provide us with an overview of Biomanufacturing 4.0. So I'll hand it over to you, Akash.
Thank you, Stuti, for the lovely introduction. Before we move ahead, and dwell into what is biomanufacturing 4.0, I would like a brief moment to introduce how the term biomanufacturing 4.0 derives itself. So, if you look at the past 200 years of human history, we do see four industrial revolutions that we have witnessed. In the early 19th century, humankind discovered steam power and then hydropower. And then in the late 19th century, we shifted to a second industrial revolution, which was, harnessing the power of electricity, and then it moved to mass production of commodities. From there, in the late 19th century, we moved towards, we see an third industrial revolution, I would say, which is the advent of electronics and IT. So, and then in the last one decade, we have seen altogether a new industrial revolution, right? Which talks more about digitization of commodities, shared data, and a data-centric industrial revolution. So this is where Industry 4.0 derives its term. In revolution, which is more centric towards data, manufacturing, which is more centered towards, which is more data-centric, which is more customer-centric, and then introduction of several new technologies. So that's what biomanufacturing 4.0 is about. It draws its premise from Industry 4.0 and uh, and integrates all the new technologies that are there. So when we talk about these new technologies that are presently that the biomanufacturing 4.0 encompasses, we see some key elements in that. One being the Internet of Things, which is very common and which is very prominent nowadays. It's there in every household, in every aspect of our lives. We are connected to the devices. Similarly, we see a very centric approach on big data. So I would say big data is somewhere where all our next manufacturing processes are driven from. It is used to leverage out loopholes, create a more centric approach, gather insights on where we can create a more cohesive and more streamlined systems. Then we see cloud computing, which is use of cloud services to study and to extrapolate to manage big data. Then in the last few years, we have also seen introduction of AR and VR, augmented and virtual reality systems, which has helped in designing in silico systems, which has helped in models data, which has helped us create systems and models which are very cost effective. We don't have to run them through any specific development phases, but in an virtual reality we can set up, it helps us design and develop these systems. And then we have also see about automation. Introduction of robotics in manufacturing has helped us reduce the hazard potential in hazardous situations, automate multiple processes wherein human intervention is not at all required. And it has given us a leverage to manufacture more precise products. Apart from these, other verticals, I would say, are simulation, integration of systems, and 3D printing. So all these new technologies, what they have given us is a platform, collective platform, to create a biomanufacturing environment which is more data oriented, more streamlined, and more cohesive. Now, if we inculcate all these inclusively, we see three to four different results here. One is the interconnectivity has improved, data transparency has improved, decentralization and decision making has happened, and customer centricity is improved. So when we talk about interconnectivity, what we are talking about is multiple independent systems have started now sharing knowledge with each other, and because of this knowledge, the bottlenecks are closing. When we talk about data transparency, it has created a more cohesive, more knowledge sharing platforms wherein each individual system is aware of what's happening within the parent system, so the decisions can be made faster. Decentralization has occurred, which has via systems and algorithms, which has helped the stakeholders to make key decisions without any backlogs. And then there is a customer centric approach. So when you talk about customer centric approach, in today's world, customer centricity has become a key important factor, whereas specific individual needs are catered to for each individual customer. And Biomanufacturing 4.0 has a key emphasis in customer centricity. Now, based on all these results, there are values that we draw out of this is efficiency, reduce manufacturing cost. As we implement these technologies, the cost of productions have gone down. We have enhanced the flexibility. We have the real-time decision making has improved and customization has improved. So this in crux is, I would say, a cliff of all the Biomanufacturing 4.0 has to offer. Now, if we go further down into understanding the Biomanufacturing 4.0, we do understand smart manufacturing triad as one of the key components of Biomanufacturing 4.0, and this again talks about automated production, automated learning, and in silico designing. So all these three components together create the smart biomanufacturing triad. Now, with the help of this triad, what we can do is we can do experimental planning, we can do data-driven modeling, as well as real-time data is being shared across the systems. So it is a continuous process, that helps us, in a continuous improvement space, helps us continuously improve the processes. Moving ahead, I would also like to, give some gist on what smart biomanufacturing, how is it bit different from from biomanufacturing 4.0. So when we talk about smart biomanufacturing, it is use of analytical tools and technologies and draw data to create more streamlined processes for biological development. Now, this would include biologicals, pharmaceuticals, as well as other biological products. And this particular system relies heavily on, high levels of measurement, precise controls, precise temperature. All this is introduced via smart biomanufacturing through integration of multiple technologies. And this is the platform where most of the focus will be in the near future. Now, using smart biotechnology also enables us to create efficient, cost-effective, and environment-friendly products, which is the need of the hour, with more and more emphasis on environment concerns and climate changes. Even the manufacturing spaces, including biomanufacturing facilities, are under some kind of scrutiny to reduce their carbon footprints, reduce their waste management, and have an efficient waste management. And smart biomanufacturing is at the crux of this, help companies innovate as well as manage their product manufacturing. Moving ahead, the second component of smart biomanufacturing, I would say, is the aspect of automated learning. Now, when we talk about automated learning, it is, as we say, it is the use of AI-based tools and to identify data patterns, to gather data from the large scale of biomanufacturing processes, and to correlate this data in a way that optimizes the entire process. Now, if we dig down, there are 4 caveats of automated learning that are widely being adopted in biomanufacturing, and that is deep learning, reinforcement learning, transfer learning, and explainable AI. Now, these 4 caveats altogether form the crux of automated learning, I would say. Now, when we talk about deep learning, it talks about gathering deeper insights from the vast data sets. When we talk about reinforcement learning, it talks about how certain AI-based systems can independently function based on the environmental feedback. So this talks more about the automation that can be implemented using AI. Then when we talk about transfer learning, it is another form of automated AI learning, wherein we can talk about systems who can implement a scale up and scale down simulations, and these AI systems can train other AI systems on how or what would be the likely outcomes. And explainable AI is something which is very new again. This is an AI system within the biomanufacturing which would talk about certain automated processes when they take certain decisions. What was the reason, what was the reason for such decisions that were taken by the automated AIs? So it would help humans understand how automated processes are making their own decisions, and what are the logics that run behind taking those decisions. That's the part of automated learning in biomanufacturing. Now, when we move ahead, the third component of smart biomanufacturing that I believe is very critical is the in silico designing aspect. In silico design has enabled developers to design biological processes, systems, and products without an intervention of a lab or a real manufacturing facility. Now, this enhances first of all, it reduces the cost to an extreme level, wherein scientists can simulate their run their simulations based on their assumptions without doing any real world experiments. It has helped to do such things. And another aspect is that it has helped to reduce costs. Now, when we talk about this, there are again 3 caveats in this as well. When we talk about how in silico designing can be implemented, we see that we can implement it using AI and machine learning platforms. to simulate new biological designs, whether how to predict the behavior of in pharmacogenomics and pharmacokinetics, and how the bioprocessing optimization can occur with certain changes in the system. When we talk, then there is another scale of multi-scale modeling, where a simulation can be run to study the scale up and scale down predictions, and accurately predict how the system would behave once if we scale up or scale down on a certain biological product development. And the third caveat is in silico based synthetic biology, where in silico designing is again very important. So since biological materials are very niche and difficult to manage and produce, a new form of synthetic biology is being developed, and in silico designing is helping to simulate how a molecule would behave, or how the molecule would bind to other biological systems, and how the components of these biological molecules or non-biological molecules will behave with biological molecules. So this is where in silico biomanufacturing is designing is helping in biomanufacturing. And these three components, I believe, are make the smart biomanufacturing as a next-gen biomanufacturing process or system. Now, moving ahead, if you talk about in nutshell, there are the processes and value additions that happen within the smart biomanufacturing would early on begin with digitalization of things, wherein we capture data digitally, digital data is captured, then we integrate this data, and integration happens wherein we need wherein we a change in structure is required, online visualization is required, and in such a way we create a digital platform. Now these digital platforms would help us in modeling and create a digital twin. Now a digital twin is itself a kind of an entire digital duplicate of the live process, which would help the stakeholders to govern and manage the entire process and simulate the entire process within a digital twin. It is connected to the it is connected to the actual system. So any changes or any anticipatory changes can be anticipated, any changes that are expected, or any I would say any feedback that would be generated from the real system can be actually anticipated in the digital twin beforehand, and this would enable the the stakeholders to manage it quickly. Then comes the management part, the smart digital stakeholders. Now, since we have created a digital twin, the stakeholders do not have to be present within the system all the time. They can remotely access the systems. They can check, make their quality check, they can make their decisions based remotely, and that gives an opportunity to have a cohesive control over the system. And with all this and post management would come the automation, which is again introduction of robots and smart factories, wherein all the other components come in together and most of the processes get automated. So this in a nutshell is the overall smart biomanufacturing, which begins with digitalization and set automation, and there is knowledge and data share across the system. So overall, this is what I had to, this is what the biomanufacturing 4.0 as a concept is. That's it from my end. I would hand it over to Stuti again. Thank you.
Thank you so much, Akash, for sharing your views on biomanufacturing 4.0. That was really informative and interesting. So now I would hand it over to Dr. Likesh to present his perspective on the topic. Over to you, Likesh.
Thank you, Stuti. Good evening, good morning, everyone. So today we are going to discuss about what from Site EY point of view, how we're going to industrialize the cell therapy ecosystem and how do we usher this biomanufacturing 4.0. So if we can go to the next slide. So in this presentation, what it will be divided into 4 sections. First section, I'll be discussing the therapeutic landscape when it comes to cell and gene therapy. Then I'll also discuss in the next section some of the roadblocks which we are facing in this manufacturing and how do we address them. And then we'll also discuss some of the detours and some of the research which we have done recently and to find this new way. And we'll end with some of the technological solutions which we have come across now recently and which we are developing within Cytiva. So move on to next slide, please. Yeah. So at the end of cell and gene therapy is the patient, right? So we have people like us who develop solutions, and then we have manufacturing companies who develop these cell therapies. But at the end of the day, what we care about is patient, and we want to see that these therapies get developed and we improve patient lives. Go to next slide. Yeah, and our vision is very simple, that we want to create a world in which the access to life-changing therapies transform human health. Next slide, please. So what are the things which we think about when we want to usher this new era? Is that we want to increase the speed to the market, because that's very important. The faster we can reach to the patients. Then we want to optimize the manufacturing process, which be suitable for this very increasingly fragmented market. We want to manufacture with agility and efficiency, and we also want to make sure this supply chain to be very robust to support this rapid growth and the shocks which we have seen in past pandemic years. Next slide. Yeah, so now we come to the now I'll discuss a little bit about what is the therapeutic landscape looks like for the cell and gene therapy. I'll go to next slide. Yeah, so now you see that there's a huge number of clinical trials going on all over the world. So that includes in Asia, Americas, Europe. Obviously, the Europe and US represent the most share, but you also see a lot of clinical trials now coming up in Asia, and few have started in the rest of the world also. And a lot of these trials are focused on immuno-oncology, because as we know, cell and gene therapy, the major target disease is immuno-oncology, but we do see a lot of interest in gene therapy and tissue engineering. So there's a huge, very strong pipeline which creates a growth for the next coming years. Next slide. So where do we see this cell and gene therapeutic landscape going? So when we started in late 2000s, it was all ad hoc process and reagents, but we are seeing that over time there is new generation of tools and therapies and processes are coming up, and we are seeing that potency of the drugs will increase, and the automation will also increase. What it will enable is from the first commercial launches and treating very few patients. Slowly the number of patients treated is going to increase. There'll be a complex supply chain and a broad clinical adoption. That's what we see in the future, and the cost will actually come down, and we will see a more globalized manufacturing and distribution. Next slide. So now cell and gene therapy, unlike the other cell therapies other therapies which we have seen in past, are much more complex. Like the initial therapies which were small molecules, which are synthetic and relatively simple. But as we are moving more and more closer to the biology, we are seeing development of MABs, which is slightly more complex and much closer to the real scenario. Then from MABs we see the cultured cell and minimally manipulated cell, and on the highest end of the complexity and synthesicity is what we see is the engineered cell. And these modalities are therefore increasingly complex and requires much more input from a synthesis point of view, from engineering point of view, and from the parameter point of view. Next slide. And also, because these things are complex, they include interdependency of biology, because obviously you're talking about cells which are living in themselves, but at the same time they have synthetic component to it which needs to be engineered. They could be derived cells. At the same time, this has to interact with the manufacturing, where you need to talk about the scalability, because every type of cell therapy will have a different way of looking at the scale, and I'll discuss some of them a little bit later. Obviously, you need to discuss from the digital point of view how this manufacturing is going to really integrate. How do we integrate new sensors, and again logistics, and how do we going to maintain the chain of custody, chain of identity, banking, tissue processing, all those things. So these new therapies involve interdependency of all these three elements, and we'll discuss these challenges as we go ahead. Next slide. So now let's discuss those roadblocks. Next slide. Uh, and this chart is actually true for almost every every therapies, but even so more prominent in case of cell therapies, that we need to start from the idea and then it leads to product, and there's a huge gap in between, right? And there's a huge investment. But between this research where we develop the new therapy and the launch of a successful commercial product which leads to customer, there's a huge development space. And the idea of Cytiva and other supporting companies is that how can we close this gap so that we can increase the time, decrease the time which is involved in the prototyping, testing, manufacturability and serviceability, process development, optimization. The idea is that how do we close this gap? That's one of the challenges which we need to address. Next slide. Yeah, and another thing is that how do we design modular and highly dynamic range expansion platforms? Because these new therapies could be T cells where the growth format could be 3D. Then we have iPSCs and lentivirus where the growth format is more 2D. Then we have a scalability on NK cells and dendritic cells where the scale is not that big. But when you talk about iPSCs and lentivirus, we can actually do scale out. So we need to think about all these systems and how do we support all of them. Because we need to talk about the control processes like DO, pH, cell density. We need to talk about the cell growth format. Then comes about bioreactor mechanism. It could be static or stir tank. We're going to do a shaker or rocker format. All these things which we need to cater, and we need to make sure that if we are going to support this new generation of cell therapies, how do we support different growth formats? Next slide. So now the challenges comes up is when we talk about how do we scale these therapies. Now therapies which are much more individual and focused on one patient, for example, autologous therapies are very different from the traditional therapies like which have mesenchymal stem cells or cardiomyocytes. The difference is that some of these therapies which are allogeneic require scale up, so you can simply increase the manufacturing scale, have a bigger and bulk manufacturing, a bigger bioreactor for that matter. But when we talk about autologous cell therapies, then we cannot do scale up because they are individual and patient specific. So what we need to do is scale out, which means that we need to have a unit operations which needs to be increased as we increase the number of patients. Next slide. Now, how do we find out new way given these challenges what we have? Next slide. So first thing is that we need to talk about all the different modalities involved in these therapies. So what are those modalities? Like you have in synthesis silico, where you need to design using bioinformatics the payload design. We need to have then once you have designed your payload, we need to have payload production, which will involve plasmid production or map production or protein production. Then we need to develop cell lines. And these cell lines needs to produce maps in case of maps or mRNA or viruses in case of lentiviruses, or we need to modify these cells to develop more advanced cell therapies like CAR-T and other types of cell therapies. So we need to keep all these modalities in mind and support it. Next slide. A second stage would be that once we have identified all the modalities, now we need to divide it into different subroutines and then support these subroutines. So when we talk about autologous, it will be selection, enrichment, engineering, cell expansion, formulation, analytics, and then finally release. Then we talk about allogeneic, then we'll focus more on the banking. And connecting all of this needs to be a digital platform which can support all these modular subroutines. Next slide. Yeah, now I'll give some of the examples that how Cytiva is doing and supporting these different modules and developing new techniques. So we have a going collaboration with CCRM. CCRM is a cooperative organization built between Cytiva and Government of Canada and Government of Ontario. It was established in 2011 with about $15 million seed funding. It has like a global network of partners with more than 95 employees, and we have a huge facility with 200 plus projects running. And I'll discuss some of these projects in context of cell and gene therapy. Next slide. So this is the kind of the facility which it really looks like in Toronto, and you can see all other partner institutes which are there, which partner with CCRM, and we help them develop cell and gene therapies. Next slide. So was one of the first things which we have done with the help of CCRM is to identify where the gaps lie. So the gaps you can clearly see lies in the automation part, where we need to think about how do we close the process. These are some of the big gaps when you talk about patient specific manufacturing. When you talk about bulk cell manufacturing, then the gap comes in terms of intensification. How do we intensify the processes, and how do we reduce the cost and make it more efficient? Next slide. So one of the examples is Bridge One, where we develop this fast track process development services, where we partner with different service providers to deliver cultural intensification, simplifying the workflow, developing new media. How do we make sure the process is closed and automated? And also we provide some of the cost analysis and risk reduction. So I'll be giving 2-3 examples of these things next. Next slide. So, for example, one of the things which we have done is we have developed more than 150 iPSC cell lines representing 15 different disease models, and given to all these academic investigators to use them in their investigations. We have done a pluripotent stem cell scale up of more than 35 billion cells, which is now focused on the scale up and the intensification. One of the challenges which I discussed earlier. Next slide. Another example is we focused on the viral vector production. Like we can go from 50 liter lentiviral vector to all the way to 500 liters, where we focused on the cGMP suspension producer cell lines, plasmid design and manufacturing. Also, we improve on the sterile filtration, getting better affinity reagents to purify the viruses, and we have our reduced process ACTA systems, our XDR systems, bioreactor systems, and then we also have our tangential flow systems and chromatography systems to purify and further polish these viral vectors. Next slide. That example is that how, so we have now covered the cell line development, the viral vector development. The third of one of the examples which we have done is how do we optimize the culture media? So we take lots and lots of candidates, do media depletion studies. We do using DOE, we do a fractional factorial to find out what are the different conditions. Then we run different iterations, and finally we come up with the top formulation. So this is sort of a chart to show you that how do we build these kind of new cultures which are optimized to produce high cell densities and high cell viabilities. Next slide. So this is one of the examples where we are using robotics to actually run different media formulation, which we came through the DOE experiments, which I discussed earlier. These are chemically defined, so that reduces your risk. And then we use a lot of analytics, which reduces the cost of this new media development, because every cell line would require a more defined and a new media. Next slide. Now in the end, once we have done all these challenges, the most important part in biomanufacturing 4.0 is that how do we make a technology-driven digital highway? Next slide. So one of the important, so you can actually look at the whole process chain or the value chain of these of the development of a therapy. So it starts from the R&D, then manufacturing, and finally into the market. And then idea of any digital platform would be to have more insights. So more insights like smart R&D, which will be more product and disease specific insights. But as you move towards closer to the patient from manufacturing to the market, slowly you get more insights on how to run effective clinical trials, how to optimize manufacturing, how to actually facilitate the patient experience, which is much better and less painful when it comes to cancer therapies. Next slide. So what we have is something called a canonical automation software, which connects all the different modules in cell therapy. So we have this whole cell therapy equipment range from via freeze, via capsule, via thaw, via freeze. So this is a cryogenic and cryopreservation workflow, and we have this automation software which can actually help monitor the logistics, help monitor the temperatures, because this is one of the highest value product, and cryopreservation is frankly speaking the least defined science. But we are coming up with these automation softwares and these devices which are plugged into the software to monitor these complex processes. Next slide. So ultimately, the pace at which this new cell and gene therapy is developing is unprecedented, and we'll have systems biology which will actually help develop these therapies for years to come. We need to partnership, like we discussed with the example of Cytiva and CCRM partnership, which will drive the progress. We need digital and analytics to improve the manufacturing and clinical outcomes, and there are no shortcuts. You need to really focus and do all the hard work, do a lot of all R&Ds to deliver the results. That's all from me. Thank you. And let us know, you contact us, whatever we can help in terms of your next step in your cell and gene therapy. I can be found at my email, which I'll put in the chat shortly.
Thank you so much, Likesh, for your presentation. That was really very insightful. So now moving on, I'll pass it on to Mr. Mario Bellman to present his take on the cell manufacturing and the challenges associated with it. Over to you, Mario.
Thank you so much. And yes, could go with for the next slide, please. Go next. Other two, please. Yeah, I decided to put this small content in the presentation, just a brief introduction. Our current situation about the cell therapy manufacturing and the challenge for us as professionals working at the cell manufacturing or cell therapy manufacturing. Next, please. Another, please. So what is the cell therapy? No, as we mentioned in the presentation before and at the beginning, talking about technology. Now we are working in directly in a life objective, no, in a life itself product, no, is the cell, but the cell in the human body, we want to repair or replace something that is wrong in the body. No, it's a technology very specific that it works on the cell or the issue or the illness that the patient have, and we want to repair the cell or repair the gene information or replace that information or that cell. No, that's the main objective that the cell therapy want. And as at the beginning, we talk about technology. Now the technology brings us these possibilities or these capabilities to move forward inside the cell, inside a good cell or a bad cell, and replace or repair. No? Next slide, please. With different technologies, with different tools, we have mainly the origin of the cells, no, where they came from. The cells sometimes or most of the times came from a healthy patient or the same patient, but another part where the cells of the body have good actions, but we want to move forward, no? So that's what I would that we talk about the autologous cells that came from the same person, and then go out and work at the in vitro environment, and then back again with the patient. No, that's the mode of autologous. But we got other options, no, from an extra donor or for a different person compatible a blood type or a cell type, and that is an allogenous donor, no, that came from an external donor, work in a laboratory to properly grow cells, get a lot of material to put in the patient, and then forward, no. And the last one is the exologist, no. There could be a different species, not a human, not the same species of the cells, but we can work on that cell therapy, modify the cells and get some products or get the cells to put in the patient, no? And we work as with a lot of information, but the reason of the therapy is just to repair, no, just to go forward with the health of the patient. But we know that the illness or the disease came from external factors or maybe for internal factors, no. We have a lot of tools or a lot of information when the illness or the infection came from the external, but what happened when our own system is against us, no? If the health of the patient is not so well, and the system is corrupted by cancer or by another bad information, gene information, so it produce ourselves or our body produce bad information, produce bad cells, and we want to remove or repair, no? So it's difficult because we know how to go forward when some external attack is against us. No, we could put more defense or more protection around us. But what happened when the infection or the ill came inside us? No, that's the reason of the cancer, of diabetes, or leukemia, or another uh, other illness that came from us. We need to repair that let me say that those cells are like a punk, no, are against the system, don't want to go of the law of the system, so we need to go to them and repair or replace, no. Next, please. So we use a lot of types of cells at this nowadays. We work with the neural cells, with mesenchymals that are the most popular. It depends on what kind of problem we want to solve or what kind of ill we want to repair is the type of the cell, no? But sometimes in some countries like Latin America, we don't have a lot of regulation, no? Internal regulation as a country or as a yes, as a factory. We don't have a lot of regulation, but it's not a normal factory. No, it's something that could fix and move forward. It's something alive that we try to fix and move forward. No, so sometimes working with human embryonic cells could be a topic of very risky topic because some countries don't have allowed this kind of experimentation or this kind of production. It is more like a ethics situation, no? But we have a lot of type of cells that we can work forward and have different tools to move forward to the reparation or removal of the cells. Yeah. Next slide, please. The second, nowadays, the cell therapy is based very based on the root of cell therapy is cell culture technology. No, we cannot move forward with the cell therapy technology if we don't have a very robust or very strong cell culture technology. For that, we know, as we mentioned in the two presentations before, that we have some tools. No, we have equipment, we have shakers, we have uh, this combination of this conjugate media, cells, and environment, no? And sometimes we want to keep most of the characteristics of the cell, no? We just want to repair one one bad situation in the cell, but we don't want to remove or change completely of the cell, no? We want to keep some information, very specific information that give us a clue or give us a power to move forward to repair or to remove the bad cells, no? So in the cell culture technology, we need to grow these cells. We try to give them the perfect environment around them just to get a lot of material. No, the next slide, please. This moment is very complicated because the raw material or the, if you let me say, the product of health is the cells, no? But if we work with just one or two cells, it doesn't work because we need a bunch, we need a lot of cells to get a good result in the body, no? But the massification of production is the clue, the point, the specific point that we need to address. We need to solve that massification of cells because sometimes, and the guys that work with cell culture or cell therapy, don't let me lie, that the raw problem or the huge problem is the material, no, or the the quantity of cells and the viability, no. If we don't have enough cells, or we have a lot of cells, but the viability or the characteristics are not properly well, uh, doesn't work the therapy, no? So we can put the cells again in the patient, but if they are not strong, or if they are a lot, not a lot, they are just a few cells, the risk or the results of therapy won't be good, no? So we want to that level of manufacturing is a topic that we talk at the beginning. No, this manufacturing 4.0 is important because we need to massification of something very small and alive, and that we need to keep that conditions. No, we need to put for the cell all the things that they want, they need to grow properly, but we need to for us as a industry, if you let me say, as a manufacturer, we need this massification, no? So we need to in small volumes, we need to keep or to maintain the conditions, but that condition we need to move forward and be able to grow as a factory, no? But those factory indeed are not so big, no, because we need to return the product to the patient to get the proper result, no? And that environment, or that universe of the cell in the media, in the carrier, in the conditions of culture, give us a lot of data, no? Give us a bunch of information that sometimes with a pen and a paper is impossible to know, no? We need to silico information. We need more technology. At the beginning, I saw a slide with this circle with different arrows pointing the manufacturing, no? And that is kind of the cell culture technology or the cell therapy technology, no? We need a lot of information of different technologies as a software or as a how is a hub to control or to get all that metadata. And sometimes it's important, no, because not only the growth of the cells or the amount of cells is important, we need to know what happened in that cell, no, and we not know we need environment control to get this proper growth of cells, no. The next slide, please. So we need to scale up, no? At the beginning, as I mentioned, we need to know the conditions of these cells. We need to a bioreactor or shaker very well controlled that give us all information that we know that we need to know about the cells, because the cells indeed are a live organism that we know that something is wrong in it, no? We change the condition, we change maybe sometimes selecting one of the good cells around the bad cells with different techniques of selection, and then go to the scale up, no? That good cell, that good information, that's what I want, no? As it's our goal. And now we want to keep and increase and get a bunch of cells that work for our patient, no? At the end, it's a tricky situation, all this bioprocess, because never we'll never forget the end of this process, no, is the patient, no, is give a solution, give health, give a protein, give a media, or give a cell that could bring solution, could bring health for someone, no. But in all that way, we need to go back and make some engineering, make some technical advantage, or make more knowledge, not so clinical, no? We need a lot of information, because sometimes the scale up for this process could bring us a lot of cells, no? But what happens if those cells are not properly in the conditions that the patient need, no? Because we need to work in a good manufacturing ambient or environment, sorry, to go forward and get a good equipment, get a good result, no? And so that give us uh, a, um, a change of how to say of goods, no? If we start with something in the process good, with media, with shakers, with environment of the cells and the cells, if we start with taking care with quality, at the end of the process, the quality of the product for sure will be the optimal, no, for the patient. And for upstream, the thing is not so hard, if you let me say. At the end, it's hard, no? But we have a lot of technology around of bioreactors or shakers or equipment or software that can give you a clue or give you a very, very good tool for get a lot of cells. But get a lot of cells is, if you let me say, is almost easy, no? Because the bioreactor and the process of upstream is attractive, is well known, and we, if you let me say, is very, very robust and constant, no? The bioreactor give us that solution. But the problem always in cell therapy or in cell manufacturing for cell therapy is the downstream, no? Because at the end, the product is indeed the cell, no? So we need to remove everything around the cell, the carrier, the media, and other things that we don't need and the patient don't need, to go down and get the pure cell, no? But this cell, we need to get that cell in a proper conditions, in a higher viability, in a lot of yield of the cells, no? So the next slide, please. So for the downstream is very complicated, no? And we have different applications after the bioreactor. Sometimes when we are working in a R&D level, is not so how to say, is not so tough, is not so um, limited the process. But when we work in a international um, how to say, regulation, we need to cover all the things that the global regulation is asking, no? And some technologies for downstream, not all technologies have a good manufacturing practice, but in bioreactors and for the scale up of for the process in the upstream, we have a lot of tools for good manufacturing, no? So in the applications, we could get the cell and we decide where to move, not to autologous vaccines against cancer, or for production of skin or cartilage, for cosmetic. Uh, at this moment, those are some applications that I know, but I are increasing. No, nowadays the meat production or in vitro meat production is it's the same technology that the cell therapy is using, no? Just for the food and beverage market. The next slide, please. So At this moment, we are talking about the next for the future. No, I used to talk with these technologies with some family or some friends outside of the biotechnology knowledge, and they told me that, oh, this is the future. No, sounds like a sci-fi movie, but indeed is the technology nowadays that we are working. But we have a lot of challenge in the future. No, not just in the bioreactor or in the robust production of massification of the production of cells, but we have a lot of things to move forward for the technology around the cell, no? The technology about carriers, the technology to keep viability after downstream process, no? After purification, because sometimes a lot of these cells need to keep the adherent conditions to grow properly and keep that function inside the body to work properly. No, if we want to produce skin or we want to produce cartilage, we need that tissue that keep the cell and don't give us another result. No, so to keep that information or that tissue information is very complicated at the downstream process. No, so that are so many challenge to get a lot of cells. It's not just the amount that we could address in the bioreactor, but we need to move forward for the downstream process. No, the next slide, please. And the problems for the upstream, the scale up is the yield. No, sometimes we have a lot of cells, but the viability or the condition of the characteristic indeed of the cell are not the proper that the patient needs. No, so we need to get more control in the environment, get some technology as mentioned in the presentation before. Sometimes um, artificial intelligence, knowing the process and get all the data and give us information about how to move forward, no, how to get a good result, but not for the in a mix with the human decision and the artificial intelligence with all that memory that could keep in uh, the batches or the process before that could move forward, no, and give us more efficient in the yield without less human error, no. But at the end, the decision of the process at this moment, and I think for the future will be part of the human, no. So what the yield is one of the challenge, and the other, as I mentioned, the harvest, no, the cell recuperation, the cell recovery, or the harvest to high viability at this moment with good conditions or with how you say the sterility and the conditions that the patient need is kind of difficult, no, but is a weird situation because when we work in a bioreactor, the environment of the bioreactor is a class, ISO class, very defined, no? But when we work in how you say in the clinical room, the environment conditions are lower than the bioreactor, no? So we are reducing the environment protection, but at the end we need to keep the cell as we need. No, sometimes in the harvest at the process we have ISO 9 conditions, but the equipment at this moment can keep that level of sterility of the conditions that the patient needs. So that's an important point to go forward, no, or like a challenge. Next slide, please. And the fill and finish, no, we know that the final product is the cell, but we need to put in a form that the patient could address. No, some of technologies or some of the therapies go to the bone marrow, no, and go inside the bone marrow for the patient is a tough situation, but we need to we need to extract the cells and go back. We need to conditions, no, this polishing and this purification of the cells is a topic that at this moment is not is not the perfect condition. No, we need to improve in the conditions that we could give the cells for the patient and reduce some future infections or problems, no? And the facilities, no? Sometimes the put a bioreactor in a facility is very easy, but have a bioreactor in a OR, in a clinical room is very difficult, no? But keep those different engineering level, a clinical level in the same building, on the same facility, sometimes is not easy or is very, very expensive, no? And that is a situation that we need to go forward for small bioreactors or very small conditions, like a lab condition, but with a highest yield or highest cell density growth to get all the conditions to move easy, no? Not, don't have a huge bioreactor close to the patient, but would be possible a small bioreactor with high density in the same building of the clinical trials, no, of the patient, no. Next, please. And in conclusions, well, at this moment we have a lot of information or a lot of technology for cell culture selection, for grow the cell types, or for selections, or bioreactors to grow the quantity of cells. But we need to move forward with different I say we need to move forward in this combination between the engineerings that they get the cells, the medicals of the clinical part give us more information about what does they need and move forward with the gene expression and all these tools that at this moment are capable to give us information about the cell and the gene and so on. But we need to get a global regulation for this kind of technology, no? And this moment uh, is not so popular around the world in the legal entities to get some laws or to get some regulation for this, no? So it's important because as a near future, this kind of therapeutics or this kind of technology will be part of our everyday for those illness or for those sick that came inside of us, no, that we cannot cover. We need to repair or replace the cells, and we need to work. At this moment, we have a lot of tools, but we need to improve, no, for a better solution in the future, no? The next slide. Yeah, thank you.
Thank you so much, Mario, for providing your valuable insights. That was really very informative. So now I would like to navigate you all through the cell and gene therapy biomanufacturing market. So what is the ecosystem of this market? What are the market dynamics, trends, and current and future market outlook? Next slide, please. So let's first talk about the market ecosystem. So there are three main end user for our market: contract research organization, life sciences companies, and contract manufacturer organization. So our market, the cell and gene therapy biomanufacturer, cater mostly to these. Life sciences companies hire CROs and CMOs to research or to manufacture their product, and then they cater back to the hospital. Next slide, please. So let's talk about the factors that are promoting the growth of cell and gene therapy by manufacturing market. So in the last few years, we have seen that there are a lot of cell and gene therapies that have been approved for commercialization. As you can see, the numbers are presented here. Along with that, we have received a lot of, we can see a lot of investment and fundings were also provided. In alone 2020, 2021, we can see the market received 22.7 billion funding. Apart from this, there are a lot of newcomers in the market. So altogether, these three factors are interconnected with each other and are dependent to drive our market. And currently, they are driving the market and are expected to boost our market in the future as well. Next slide, please. So now talking about the key trend, as Akash and Dr. Likesh has already told us about the biomanufacturing 4.0. It's a new big thing in the cell and gene therapy biomanufacturing. There are other few key trends that have been adopted by the industry or the key players to mitigate the risk or to produce the efficient therapies. So one of them is the growing use of single-use technology market penetration. So with the help of this, we can the players can obviously reduce the financial risk as well as can reduce the chances of cross contaminations also, and it is cost effective too. Next slide. So apart from that, there is also a shift to close processing biomanufacturing. As Mario has told us, the process of cell manufacturing and the challenges associated with them. So the biggest challenge in the production of cell and gene therapy is the chances of cross contamination and the handling errors. So with the help of close processing manufacturing, we can eliminate this risk and can have a better the cell and gene therapy developers can create more effective therapies. Next slide. So there are a lot of new opportunities for this market also. So one of the biggest opportunity is the presence of robust clinical pipeline. So there are a number of clinical trials that are going on that would help, that are currently helping, and when once they get approved or reach to the stage of commercialization, will create opportunity for our market. Next slide. So currently, biomanufacturing market is a very vast market. It consists of monoclonal antibody, cell and gene therapy, drug antibody conjugates, etc., etc. So, but our main focus is cell and gene therapy, which holds approximately 20% of the market. Next slide, please. So, according to our analysis, or according to, in our opinion, currently the cell and gene therapy biomanufacturing market holds is estimated to be 11.65 billion and is expected to grow to 29.76 billion in 2031 with a CAGR of 10.31% in the forecast period of 2022 to 2031. So we have also segmented this market according to the product type, which are consumable, equipment and software solutions, and currently consumable holds the largest share of this market, and this trend is anticipated to grow since consumables are used since the equipments are capital investment and have a shelf life of 6 to 7 years, but consumables are required each time when a therapy is used or created. Next slide, please. So after this, currently according to the scenario, as we have presented earlier also, a lot of therapies are in research stage, are in clinical pipeline, and are not commercialized. So currently the trend is that that commercial stage manufacturing holds the larger market share in this market, and this trend is anticipated to shift towards the I'm really sorry, research stage manufacturing holds the largest market share right now, and as we move on, the trend is anticipated to shift towards commercial stage manufacturing, as these therapies that are in currently in clinical pipeline would in next 2 to 3 years will get approval and get commercialized. So this trend will shift and the share of commercial stage manufacturing will increase with time. So that's all from my end. Now with this I'll conclude today's session. So we at BIS Research are committed to building a repository pertaining to cell and gene therapy. So currently we have multiple reports published and we have several other in the pipeline. So here are some of the titles that I would like to highlight. Next slide, please. So now I would open the panel for any question from your end. You can type in the questions. You can mention the name of the speaker that you want to direct your question to. And before that, I would also like to talk about InsightMonk. So InsightMonk is a proprietary platform created by BIS Research to connect with the KOLs. You can sign up there as a user or as an expert and impart your knowledge, share your knowledge with the other people. So through this platform, you can get access to the market reports, PDFs, and databases of key players. So now I would just take the questions from the audience. Just a second. So this question is directed toward Likesh and Akash. So could any AI program predict, modelize cost effectiveness of a new therapy or device in a particular country, or AI as market access support? Either of you can take it. Likesh or Akash, either of you can take this question.
Hi. Yeah, I mean, it's Likesh here. So yeah, I mean, in principle, yes, but I mean, I would say the question is too broad. Yes, it can predict, but then you need to define a lot of parameters, like how, like, if you want to predict a particular drug, how would or particular device, how to do in country, you need to get a lot of data, which currently I don't think it exists. So I think the limitation currently is not with the tools, but with the data. But yeah, in principle, it is possible.
Thank you, Likesh, for providing the answer. I'll check for another question. Just give me a second. So the question is, who are the main software vendors in this ecosystem? It is not directed toward anyone specific, so either of you can take it. Mario, Likesh, or maybe Akash, anyone.
Well, for our point of view, from Inforce, we develop software for managing all the metadata that you produce in the bioreactor, no, or in the culture or shaker. So we can get a lot of information and move forward as a recipe. No, keep that information, put a recipe, and go forward to the project. No, indeed, it helps you a lot in this cell culture, because if you have a golden batch or this very specific information that you want to repeat lot by lot, the software could help you to keep that information and reproduce the time that you want in the bioreactor, no, for have the same results in the culture.
And same from Cytiva point of view, we do have software in this ecosystem, like Chronica software, which I alluded to in my presentation, which takes care of all the batch manufacturing record from end to end, and it also supports in logistics and inventory management.
So, as we are very short on time, I'll just pick one more question, and then we can answer the rest of the question through blog post. So the next question is, are there any companies working on cell and gene therapy in India? So again, it is not directed towards anyone. So either of you can pick up this question.
Akash, would you want to take it?
Yeah. Am I audible?
Yeah, you are audible.
Yeah. Currently in India, I would say the scenario is more towards stem cell development. There are few companies who are working in developing cell and gene therapy. From the top of my mind, I can say three companies do come to my mind. One is Immunel, one is Intas Therapeutics, and Stemputics. I guess Stemputics is a Bangalore-based organization. So these companies are trying to create cell and gene therapies. However, they are still in development phase because, as our speakers have rightly pointed out, right, that it is a very capital intensive as well as a very lengthy process. But yeah, we do believe that in next decade or so, we may have some kind of cell therapies or gene therapies in Indian market.
So yeah, as I have already mentioned, we are running short on time. So we'll just conclude this session with this question. For the rest of the questions in the chat, we'll answer through the blog post. So yeah, that's it. Thank you so much everyone for joining in today. Thank you Likesh, Mario, and Akash for providing your insights on the topic. So yeah, thank you very much for joining us today. Okay, so yeah, have a nice day everyone.
Bye. Have a great day, everyone.
Thank you. Bye-bye.
Bye.